I think that we may start the session for today. So hello, the dear attendees. My name is, as you know, Yasin Samawet Shafiq, and I've been your host for the last month. And welcome to one of the sessions that I've been most eager to host in this US SynBio Space Festival. So today we've got Lucas Boldrini, who is a technical consultant at the International Biosecurity and and Biosafety Initiative for Science, IBBIS, as you may know. And he's also the founder of the iGEM Space Initiative. He is working at the intersection of biosecurity, synthetic biology, and space governance. Lucas works to advance biological safety standards and the international policy frameworks for emerging space technologies. He previously managed global operations for over 400 teams at the iGEM Foundation and represented the community at the UN Committee of Peaceful Uses of Outer Space, COPUOS. Lucas holds a master's in biophysics from the Barcelona Institute of Science and Technology, with research experience spanning stratospheric experimentation, neurobiology, and biotechnology. Thank you so much for being here, Lucas, today. As a fun fact, Lucas was the man one of the men who started my spark on space synthetic biology. So that's why I am extremely happy to having him here today, talking about space policy and how we can navigate the problematics and the frameworks that we're going to see in the next years with space exploration. So, Lucas, the floor is all yours.
Hi, Yasin. First of all, thank you so much for the invitation. Thank you, everybody, for being here. I will probably start maybe in a minute or two so that potentially a couple of other people may join. But in the meantime, I just have to emphasize again that I'm very happy that you, Yasin, and the EU SynBio Society are both hosting the Space Festival. It's so synthetic biology applied to space exploration is a topic that is not widely discussed around the world. And it's an intersection that is often more often than not overlooked. So there's a bunch of people working with synthetic biology, a bunch of people working in biotechnology, and a bunch of people working on the engineering side of things for space exploration, for aerospace technologies, and then it's very hard to basically marry those two fields together and blend them in. So thank you for actually providing the platform to do that. So okay, I will start then sharing my screen, and hopefully you will see it. Yasin, could you please give me a thumbs up if you can see my screen?
Yes, we can. We can see it properly.
Okay, perfect. So yeah, today we're going to discuss a little bit about the interplanetary guardrails, so space law, biosecurity, and frontier tech governance. And it's going to, like, I already warn you that it's kind of heavy in content. So I'm sorry if you're going to be a little bit overwhelmed at first, but I promise you that in the end, everything is going to make sense. So at first things first, like, who am I? What happened to me? Where do I come from? So I am originally from Brazil with a Hungarian passport. I'm a dual citizen. It's a long story, but I was born and raised in Brazil, and I've been living in Paris since 2022. And ever since I was a little kid, I absolutely loved things that fly. So I was crazy about airplanes, about rockets, about birds, with a very notable exception of mosquitoes. I absolutely hate mosquitoes. But other than that, ever since I was a little baby, a little boy, I love things about astronomy, everything that was related to space exploration and aviation aeronautics in general. And this is something that I was born with. So every time I did something with my work, I tried to just put it inside what I was doing, like imbuing it with my activities. So I studied biotechnology at the University of São Carlos in Brazil, and my first ever lab experience was under Otávio Timan's supervision in a structural biology lab, absolutely nothing to do with space, but this was the very first time that I was pipetting, that I was doing PCR, SDS page, electrophoresis, and it gave me a lot of hands-on experience to understand a little bit more about the molecular biology world. But then, while I was a student, I was also involved with a student group from the University of São Paulo called Zenith Aerospace, where we built and operated high altitude probes. So this was one of the coolest things I've ever done in my life. I was the scientific department coordinator there. So I was mostly in charge of developing and operating and analyzing the experiments going on board the stratospheric balloons. They were mostly biological. And again, this is super, super fun. I was so involved with it that I was also helping build the probes, assemble them, launch them, recover them, and doing also flight simulations because, again, it was super, super fun. We had the scientific front for this and the educational front, where we just boarded a bunch of high school and middle school experiments from all over Latin America into the probes and send it to the stratosphere and make a competition that is nowadays across all Latin America. So this is all the team. That was, again, one of the coolest things I've ever done in my life. I cannot emphasize this enough. So briefly talking a little bit about what I did as an experiment, the very first one that we prepared was an epigenetic assay validation of the protozoan Naglera gruberi, which is the benign cousin of Naglera fowleri, the brain-eating amoeba, which is one of the most lethal diseases ever. So we wanted to basically understand how the stratospheric environment affected the gene expression of cells. And then we were just using Naglera gruberi as sort of a validation for a specific organism to be as the chassis or the platform for this sort of experiment. And we had mixed results. Basically, we discovered that Naglera gruberi was not resistant to the ultraviolet radiation that is harsh in the stratosphere, but was able to survive the other parameters in a way. The other one that was a very cool experiment of ours as well was an initial assay for terraforming. So we got some cyanobacteria that were originally isolated from the campus of our university called Aphrodite, and then we decided to just send them to the stratosphere because, well, they produce oxygen, they fix nitrogen, so they would be great, for example, to produce a habitable environment or atmosphere on Mars, for example, or in some other celestial body. So first we need to understand if they are resistant to the environment we want to decide to send and colonize, right? Outside of Earth. And the stratosphere is treated as an analogous environment to the Martian surface because it is very similar to the amount of ultraviolet radiation, low pressure, low temperature, and low humidity. So it's much cheaper sending something to the stratosphere than to Mars. In any case, we sent the Aphrodite a couple of times to the stratosphere, and we discovered that actually some cells did survive. So the ultimate plan for that was to conduct a directed evolution of the cells that resisted the stratospheric environment, and then basically creating the Hulk, ultimately making it very resistant to this environment. But then COVID came, and we kind of interrupted the experiment. The last experiment that it's important for me to mention is my iGEM team. So in 2019, I was part of the São Carlos, Brazil iGEM project, where we were developing a Saccharomyces cerevisiae, so the baker yeast, to be resistant against ultraviolet radiation to produce bread, ethanol, beer, and wine to astronauts on Mars. And then we were doing this by using melanin, using specific chassis with an yeast display system. And of course, we have a lot of biosafety concerns here. So we developed a kill switch logic gate because basically whenever the yeast, for example, escaped the bioreactor, we would just let it kill itself. So I'm not going to dive deep into all the details here, but it was a NAND logic gate. So whenever there was a lack of methionine or glucose in the medium, the cell would basically kill itself. It would just trim all of its DNA internally. And this was the image of our iGEM experiment aboard our probe in the stratosphere. This picture became quite famous within the iGEM community. So it's one of my little babies as well. And also here, big shout out to my iGEM team members. I think potentially maybe some of them are present here. So thank you again for everything that you've done for this project. It was super, super cool. Then as an interim, before I talked about more space stuff, I was briefly a research visitor at EMBL, the European Molecular Biology Laboratory in Rome, doing a little bit of work on on neurobiology. and just studying mouse behavior in their brains, territorial behaviors, aggression, and everything. That was super, super interesting. And then I did my master's in Barcelona at the Institute for Bioengineering of Catalonia, where I was studying protein aggregation in Huntington's disease. In a nutshell, that's nothing. It has nothing to do with space, but just giving a context of how I ended up at iGEM. So then in 2022, I finally moved to Paris and became team experience manager at the iGEM Foundation. So my main role was actually helping teams out conduct their registration, basically having them making sure that they have a good experience within the competition. But then, as a side project, yes, I founded with Vinu here the VP of Technology at the time, the iGEM Space Initiative, which was basically a platform to develop the intersection between synthetic biology and space exploration. And then I think the main locomotive of the initiative was the iGEM Space Network, which is a group of experts in the field of at least space biology, some with SynBio, but not many people focus on the intersection. So they're mostly experts in life sciences applied to space exploration. And then the main stakeholders here that are very important to give a shout out are firstly ESA, the European Space Agency. We strongly collaborated with them. They are amazing, incredible. Then with NASA, with Ames Research Center, with the lab of Lynn Rothschild, we did a lot of stuff with them. CNES, the French Space Agency, was involved. DLR, the German Space Agency, and then some other academics from Cornell, the University of Edinburgh, Concordia University in Canada, the Astrobiology Center in Madrid in Spain, and the University of Tor Vergata in Rome, among others, but those I think are the main ones. And also, in parallel, we created the Space Village. So a village in the iGEM competition is basically, let's say, a broad theme of the project that the teams are conducting. So we cluster them together into specific topics. So we have diagnostics, infectious diseases, oncology, software and AI, and then we develop space. So first, back when I proposed this in 2023, there were not enough teams. I think there were 2 or 3 teams. Then in 2024, we finally had a full space village. So this is the one in the picture here with 9 teams, and that was amazing. Then 2025 was 12 teams. And then this year, I recently took a look at how many space teams we have, and there are 17 iGEM space teams. And this is something that is unprecedented. So I'm very, very happy that this is happening, that we were able to incite this space culture within the iGEM community. So here we have maybe, 200-300 individuals working directly on synthetic biology applied to space exploration. So this is just absolutely amazing. And then now the star of the show is Yassine. So our host today, he is the current space mayor. He was a member of the Space Network as well. And now everything that you want to know about the iGEM Space Initiative, the iGEM Space Network, the Space Village, go talk to Yassine. He's the star of the show at the moment. And then earlier last year, I moved to IBBIS. So briefly, I'm going to explain what IBBIS does, which is mostly actually not focused on space exploration. And then we're going to go back to space. So the International Biosecurity and Biosafety Initiative for Science is a Swiss nonprofit foundation with the main goal of safeguarding biotechnology for safe and responsible advancement. So basically, we want to make sure that nefarious users don't get their hands on dangerous biotechnology materials or technologies in general. So we work with global partners to strengthen biosecurity norms and build innovative tools, trying to reduce the risk of catastrophic events from accidental or deliberate misuse of biotechnology. And currently, our main focus is on DNA synthesis technologies. So just contextualizing why synthetic DNA is important to the world currently, and then what we do to safeguard this. So synthetic DNA is inherently a dual-use technology. So first off, there's a lot of benefits for this. Whenever you're a researcher, you can do a lot of gene editing in synthetic biology applications by using synthetic DNA. So instead of, for example, you want to use a specific gene from an organism to conduct your research, sometimes it is easier for you to just ask a company to synthesize it chemically, the gene, and then put it in your bacteria to do whatever you want, rather than just getting that organism, extracting the gene from them, and then inserting and cloning into your chassis, your cells. So it is extremely, extremely useful. For medicine as well, we can do diagnostics with it. So during COVID, for example, we had qPCR, which uses primers, so short oligos that are synthetically made to detect the viruses, and of course vaccines. So a great example here are the Moderna and Pfizer vaccines that were developed during the COVID pandemic as well to tackle SARS-CoV-2 infections. So these are just a few examples. Then we can do manufacturing, so we can do genetic modification of plants, and we can do bioreactors to conduct biochemical experiments and creating products. So the industrial, for example, production of insulin, for example, heavily relies on synthetic DNA. And currently, we have now more frontier emerging fields of data storage and AI. So there are already some companies that basically their product is selling data storage information in DNA. So if you don't want to, keep a piece of information in a hard disk or the cloud, you can ask them to synthesize the DNA that codes for that information that you want to store. So it can be a video. After that, it's translated into digital language. It can be a video. It can be pictures. It can be code. It can be whatever you think about. Then, of course, synthesis screening can prevent misuse. So again, synthetic DNA is a dual-use technology inherently because you can either synthesize a gene that can produce insulin, for example, and help millions and millions of people, or you can use synthetic DNA to synthesize botulinum toxin or anthrax precursors or virulence factors of a specific pathogen. So there's not really a difference there when it comes to the technical aspects of it. It's as easy as to synthesize insulin as it is as easy to synthesize botulinum toxin. So there are two sides of the synthesis screening that can prevent this sort of misuse. The first one is sequence screening. So it's basically recognizing potentially risky sequences toxins. So whenever the company receives the order, that we need to make sure that that sequence specifically of DNA that is being ordered is not dangerous. And then the other side is the customer screening, making sure that the person who is ordering that DNA is a legitimate customer. So it's somebody who is a legitimate researcher or from a specific company, rather than just some DIY guy or some bioterrorist who wants to kill their colleagues or something. So basically, whenever you're a company that synthesizes DNA, this is what you receive. Imagine that I'm a researcher and I order this sequence. This is what they get. There's not a single soul on the planet and in the history of the world, including Nobel laureates or anybody else, that can, with their naked eye, understand what this is. There's nobody who can do this. So there's no way for us to know if this is insulin, if this is botulinum toxin, or something that is completely meaningless that doesn't code for any protein. So the sequence screening aspect of it is what we focus at IBBIS to some extent with our tool, which is a common mechanism, COMIC for short, where we basically translate what this sequence means. And then here we can see that this is a virulence factor of Ebola virus. So it's absolutely dangerous. And we should, if somebody is asking for this, we should make sure that they have a legitimate usage for that they have the proper licenses, that they have the proper infrastructure in the lab to do this. And then going a little back now to the current space-related work that we are doing, this is very embryonic. We're still, starting these conversations within IBBIS and to the outside world, where we are currently focused on biosafety and biosecurity considerations for lunar habitats. So we'll talk a little bit more about this at the last part of the talk. And then the other thing that I'm currently personally involved with is the Space Life Science European Research Network, CERN, which is a network of about 40 or 50 researchers across Europe focused on biotechnology and space biology applications for space as well. So I'm mostly involved with the SynBio part, the synthetic biology part, and the educational part as well. So now let's dive into why biology goes to space. So why is it important to focus on the intersection between biotechnology, SynBio, and space exploration? Why is it useful? So there are many topics here that we discussed today. The European Space Agency a few years ago listed 4 main areas that are the most imminent ones that synthetic biology could be applied for. So There are others that are a little bit more within the realms of science fiction right now. So terraforming, for example, that I mentioned a little bit briefly, which is basically changing a celestial body, a planet, to look like the Earth in its atmosphere and temperature, mostly, and pressure. But right now, in the immediate decades to come, these are the main four ones. So the first one is insider resource utilization. So it is basically mining things from lunar and Martian regolith to produce and manufacture things. So one thing that we could think about is, for example, microbes mining regolith both on the moon and on Mars to make cement, glass, metal, ceramics, and even solar panels. Both lunar and Martian regoliths are very rich in silicon, in calcium, in iron, in magnesium, in aluminium, which are all essential elements to manufacture cement, for example. So we can make this with microbes that we could potentially save a lot of trouble and a lot of money instead of doing this on a chemical and industrial level with only machines and chemical processes. And in some studies by ESA, it is said that we can save up to 85% of mass compared to launches. So instead of sending cement from Earth to the moon and Mars, we can just make it there, and we would save a lot of fuel, a lot of launches with this. But there, of course, there are risks. So if you just engineered organisms and just let it loose on the moon or Mars, we just don't know what can happen. We just let it loose in an environment we don't understand, and then we can possibly contaminate sites of scientific value. So we can end up having, for example, false positives when it comes to detecting extraterrestrial microbial life. So these are specific risks that are very hard to measure. We're going to talk a little bit more about this. The other one is bioregenerative life support. So one thing that we could do is that we could engineer cyanobacteria, algae, or plants to form closed loops within the habitats, recycling waste into food, regenerating oxygen, and purifying water. So this image here is a bioreactor with algae it's currently in the International Space Station, that was conducting some experiments to recycle oxygen and using the waste produced by the crew to produce a little bit of food and substrates as well for them to consume. Of course, risks associated here are that a seal habitat is a closed ecosystem, so there's no dilution and no reset. So there's a diversity loss with all the microbiome that is inside this habitat. Opportunist organisms may be artificially selected with this resistance selection as well. There's biofouling within the cooling systems, within the ventilation systems. So the International Space Station, for example, is a semi-closed loop because there's a lot of resupply missions. It is relatively easy sending crews and supplies to the ISS on a recurrent basis. But then if we go deep into space, let's say to the moon or Mars or even further, then of course the space missions have to be increasingly more autonomous. The other one is radiation protection. So basically we need to make sure that the organisms that we send to the moon or Mars will survive in the harsh conditions of that environment. So we need to make sure that they survive on the lunar and Martian surfaces that can be, the moon, for example, can be extremely hot or cold depending on the sunlight. It has no pressure whatsoever. It has no atmosphere. The radiation can be very high, and obviously we need to make sure that this is properly addressed. Same for Mars, but on a smaller scale, but still it's a very, very harsh environment. So here on the left, this is just a very illustrative image I like from a group of Brazilian astrobiologists that send these groups of organisms into the stratosphere, and then some were able to survive the stratosphere and some were not. So we can see, for example, that the fungus Exophiala and C. friedmani were able to kind of pretty well endure and survive the stratospheric environment, while, for example, even the famous Deinococcus radiodurans was not able to survive the synergistic effects of the stratosphere. So just as a curiosity, Deinococcus can survive a strong amount of radiation, but whenever you mix it with the low humidity, it cannot survive. So the risks here, of course, is that strains engineered to survive extremes are the hardest ones to sterilize and contain. They just can become too hard to control. One example that is a little bit kind of tangent, but it's illustrative, is, for example, antimicrobial resistance. We just give a lot of antibiotics to patients, and then ultimately we artificially select their microbiome into making sure that some of the pathogenic bacteria just become super, super resistant to antibiotics. So this is, in a nutshell, kind of what can happen when we just make something that is really resistant, and we don't know what can happen with this afterwards. The final one that I want to raise is the crew health. So engineered yeast as an on-demand pharmacy, so we could potentially have yeast cells basically producing medicines on demand. So instead of just bringing any drugs, to legal drugs, of course, like medicinal drugs, to Mars, to the moon, we just have them being synthesized in situ. So we can also have biosensor microbes in the gut to monitor astronaut health in near real time, to make sure that their gut microbiota is well and everything is good. And of course, the risks are that any platform that synthesizes chemicals is inherently dual use. So they could also be used to synthesize harmful substances too. So this is one thing we have to keep in mind. Now, there are three concepts, three disciplines that are important to split and define so that we can proceed with the rest of the talk. So the first one is biosafety, the second is biosecurity, and the third is planetary protection. So going to biosafety, this is mostly protecting people from biology. So whenever you are a researcher in a lab, we need to make sure that all of the equipment that you are handling protects you from, for example, being contaminated with your sample if it's a pathogen. So let's say you are a researcher, an extreme here, you're a researcher in Ebola virus. We need to make sure that you are not going to get infected. So there are several very, very characterized protocols for this on Earth, which goes from biosafety levels and everything. So this is quite established in many, many places on Earth. The other one is biosecurity, which is protecting biology from misuse. So making sure that people do not misuse biology to harm other people. So it's not about the person being infected with what they're handling or being contaminated. It's about them not deliberately or accidentally releasing it outside of the lab and hurting somebody. So it's more of I think, an ethical perspective in the sense, although there's the accidental aspect of it as well. The third one that is very important to bring up here is planetary protection, which is preventing contamination between worlds. So, for example, making sure that we don't bring microbes to Mars and then let them just live on Mars and contaminate the pristine environment that Mars is, for example. Disclaimer, planetary protection is not planetary defense. So planetary defense is whenever there's an asteroid or comet that is about to collide with Earth, and then we want to deflect it. This is planetary defense. Planetary protection is about contaminating other celestial bodies. It has nothing to do with planetary defense. And now let's zoom out a little bit and go to the space governance aspect. So right now here, this is not really, it has nothing really to do with biology or synthetic biology. This is mostly a way to understand that we're not starting from scratch, and there are some frameworks in place when it comes to how we should use outer space. So the main body that governs this is the United Nations. So it's the Office for Outer Space Affairs. The Secretariat is based in Vienna, in Austria. And then there are mainly 5 treaties that are legally binding, and they govern all of the use of space, of outer space, by spacefaring nations. So the main one is the Outer Space Treaties, signed in 1967. That is the foundation. It's basically the constitution of how nations should use space. And then the most relevant part when it comes to biology and more specifically planetary protection is Article 9, where it says that states must avoid harmful contamination of celestial bodies and adverse changes to Earth from returning extraterrestrial matter. And then also, just for out of curiosity, the other 4 treaties are the rescue agreement. So whenever, for example, there's a Chinese astronaut in distress, in danger, Russia or the United States should provide support to rescue that astronaut, or even if it's a specific equipment as well that is uncrewed, unmanned. The other one, the third one, is the liability convention, which is basically, let's say that a satellite crashes on somebody's house. So who's going to be liable for this? Who's going to pay for the damages? So this convention delineates that the operator, the nation that is operating the satellite, is the one responsible for this. So there's only one claim that has been filed so far, which was a Soviet satellite, Cosmos 954, that that exploded during reentry after a malfunction in orbit, and it was carrying nuclear material, and then some debris fell in the northern area of Canada, close to the Great Slave Lake. And then Canada filed this claim to bill the Soviet Union, and they ended up getting $3 million Canadian dollars for all the cleanup that they had to do. It's just a, I digress a little bit, but it's a fun story, not for the Canadians, but it's interesting to know that we are somewhat protected in a sense. There's a registration convention which stipulates that every object that is launched into space should be registered and communicated to the United Nations so that everybody knows what is being launched. And everything is there in low Earth orbit, or at least in Earth orbit or in deep space. And then the last one is the Moon Treaty, and this one is effectively useless because there are only, as far as I know, 17 signatories, and none of are major spacefaring nations. This treaty is not legally binding because countries did not adopt it. But it is mostly a zoomed-in version of the Outer Space Treaty focused on the moon, with the important addition of resource utilization, which is something that the Outer Space Treaty says that no nation can claim any land on the moon or any other celestial body. Much like Antarctica, it is international water, so to speak. The Moon Treaty, however, emphasizes that whenever let's say the United States mines water on the moon, that water is basically a heritage of the world and not from the United States. So that's, I believe, why most countries did not adopt it, because right now in practice, if you do this, the water is going to be basically the heritage of somebody of the body that mined it. Then within the United Nations Office for Outer Space Affairs, UNUSA, there's COPUOS, which is the Committee on the Peaceful Uses of Outer Space, which is the UN committee where spacefaring nations negotiate the rules of all of these treaties and communicate what they're doing to the world when it comes to space activities. It is a quite important forum in the space policy, in the space law world. And I've been there 2 times. I represented IGEM twice at COPUOS in 2023 and 2024. And from what I could tell, and from my own research, and that's kind of obvious, is that life sciences are mostly absent in conversation. So there are some, technical presentations. They're very in the fringe of everything, but nobody went to the main session and specifically spoke about introducing space biology or biotechnology frameworks to have countries think about laws and governances to govern this in the space world to some extent. So my little contribution to this was that in 2024 I was able to represent IGEM with a statement in the main session of the main committee, basically talking about the IBBIS Space Initiative and the importance and risks of synthetic biology applications for space exploration. So that's one of the most important things I think I've done in my career. And here's me giving the giving the statement in front of probably 300 people from 120 countries. And this is me just talking to the director of UNUSA, RT Holamaini. And so this is my little contribution to this wide world that is the space world. Okay, now let's focus on planetary protection again and just dive deeper a little bit when it comes to that. So remember that I mentioned about the Outer Space Treaty Article 9, where they say, do not contaminate other planets with earthly microbes and do not contaminate Earth with extraterrestrial matter. So there are basically two sides of this. So there is forward contamination, which is, hey, don't seed other worlds with Earth life. And then backward contamination is, don't bring anything back to our biosphere. So yes, it is very likely or possible that on Mars or on Europa or even Enceladus, there are microbial life forms, either fossilized or living. We don't know. There are ice caps on Mars. There used to be canyons, so we know there used to be liquid water on the surface. So it is possible that there's something there. And we hope to discover this sooner than later. And we just need to make sure that we just don't bring it back to Earth because, again, we have no idea what can happen. Probably nothing, but what if? So this is extremely, extremely important for both sides. Forward contamination so that we do not contaminate another planet and basically harm that environment, mostly for scientific and ethical purposes, and backward contamination because we don't know what this is going to do to the biosphere on the planet. And then the main framework that governs planetary protection protocols is the one developed by COSPAR. So the COSPAR Planetary Protection Policy, which is quite well established and it's quite well codified, and then into 5 categories. So the first one is whenever you have a space mission, you need to classify them into one of these categories. So the first one is category 1, which is no concern. So it's mostly a flyby, an orbiter, or lander to a place that is extremely unlikely to have or harbor life as we know it. So asteroids, Io, for example, the moon of Jupiter, some other, Triton, the moon of Neptune, with a lot of geysers and like excruciatingly cold temperatures that is just absolutely unlikely to have something there. The category 2 is the basic cares that we need to take, which can be with flybys, orbiters, or landers. And then this goes to places where having life is very, very unlikely, but not as unlikely as, for example, an asteroid or Io, or like volcanic moons that are, again, with excruciatingly high or low temperatures. So here the moon has its subdivision. So there's category 2, which is just a flyby or an orbiter. If there's a landing on the moon, it can be either category 2A or B. So A is whenever you are landing anywhere on the moon except a permanently shadowed region, so a crater that is always in the shadow, or the polar regions. And then 2B is whenever you go to the polar regions or these permanently shadowed regions. But then, of course, there are the gas giants, Neptune, Jupiter, Saturn, Uranus, Venus, Mercury, all of those. And then category 3 is an enhanced safety procedure focus on flybys and orbiters. So here we focus on Mars, Europa, or Enceladus, which there is some likelihood that we can find extraterrestrial microbial life on the surface of these celestial bodies. Category 4 is strict, so this is a lander that goes to these places, to, for example, Mars, Europa, or Enceladus, and then this is divided into, Mars is subdivided into also other categories, so categories 4A, B, and C. So category A is somewhere on Mars that is not scientifically, let's say, significant, that is called a Martian Mars, a region of interest. B is, I think, one of those. And then, but when you go to that, but then you're not focused on astrobiological experiments. And then C is when you go to these Martian regions of interest that have a higher potential for harboring life in specific craters and maybe canyons, or even mostly craters, or even the poles, because they have ice caps there. And then C is when you go there and you're focused on astrobiological research. And then on the other side is category 5, whenever you have sample return missions. So let's say you go to Mars and then you bring something back. This is something that we may do in the next few decades, or let's say the Apollo missions or the Artemis missions, whenever we go to the moon and bring something back, like lunar rocks, for example. So the first one is the unrestricted Earth return. So there's no indigenous life expected. So there's a simple protocol to follow. And then there's a restricted Earth return where possible indigenous life is present. So mostly from Mars or Europa, for example. And then this requires a higher amount of time for quarantine, more thorough analysis, and isolation, and so on and so forth. Then, now we focus on space biosafety, which is trying to understand what are the dangers when it comes to human crews basically operating or handling biological materials in space, right? So Earth biosafety takes gravity for granted. So all of the biosafety protocols that we have on Earth were developed with the very obvious fact that we have gravity on Earth. So whenever you go to microgravity, the basics just break. They absolutely stop working. For example, aerosols don't settle. They so like if you have a virus that is within let's say a probe or a crewed vehicle, it's just going to linger in the air for a long time. There is no natural convection, so we need to do it by forced heat or gas dispersion. For example, whenever astronauts sleep in microgravity, they need to have a small fan close to their face so that they can just put away all of the carbon dioxide that they exhale, because otherwise they will just inhale it back because it just lingers around them. So also this has severe implications for aerosol multiplication, and yeah, it's quite tricky to figure this out when it comes to microbial life forms. And liquids are floating blobs. So if there is a spillage, as we can see in the image here, that is basically surface tension is the only force that governs the shape and the motion and the interaction with the environment of the liquid. So whenever there's a spillage, we don't know where it's going to end up. It doesn't fall. So it can end up anywhere, and that's one of the main issues as well. So the other very important thing here to mention is that microorganisms may not behave the same way. So we have evidence that both pathogens, human pathogens, can either increase or decrease virulence. So the main example that I have for an increased virulence is salmonella. So there was a paper that was published in PNAS in 2007 where they flew salmonella in the space shuttle, and they found that there was an altered gene expression within the bacteria of 73 genes that changed their pattern of expression by just being exposed to microgravity, which is insane. It's very hard to measure how they're going to behave. And they increase virulence. Whenever they did some assays with mice, they figured that the strains that flew in microgravity were killing mice quicker and in a more aggressive ways that the control ones. So this can genuinely happen. And the other one that I like to bring up here is thick biofilm formation, which has been extensively already observed at the ISS, International Space Station. It can create biofouling, so the microbes accumulate in the ventilation systems, in the shafts, in some other aspects, and yeah, so there again, they can be selected again at the ISS, at the very actually the external part of the modules of the space station. They have already isolated microorganisms that were able to grow and evolve from that environment. So again, it's a huge unknown, and then the protocols and the risk assessments that we do on Earth may not be sufficient to just predict everything that can happen when we go to microgravity. And the other important factor here is that a deep space habitat is a closed ecosystem. So imagine just closing your bedroom for 3-4 years, and that's it, you know. So opportunist organisms can dominate. There's can be some artificial selection. We don't know what can happen. The crew immunity decreases. The horizontal gene transfer may increase among organisms. So given that there's a lot of accumulation, a physical accumulation of cells, horizontal gene transfer can increase. And then big disclaimer here that the International Space Station and Tiangong, which is a Chinese space station, are only semi-closed, again, because there is a lot of resupply missions that go periodically from Earth. It's relatively accessible going to low Earth orbit and keeping them, you know, having this reset every once in a while in the whole, let's say, ecosystem that they have. And again, fun fact, if you've never seen how the Tiangong space station, the Chinese one, looks like, that's how it looks like. It is actually much cleaner and less clunky than the ISS. And then now we finally go to lunar habitats, and this is the final sprint of the talk. Thank you for bearing with me. And now let's focus on lunar habitats. So this is all where biosafety, biosecurity, and planetary protection converge. So lunar habitats are closed habitats. So again, the crew lives in a confined environment, as we have extensively discussed here before. Now, a big aspect of just having a moon base is scientific purposes. We need to do research there and then use this as a stage post to then colonize other places in the solar system, mostly Mars, but it's mostly for scientific purposes. So it is expected that, like in the International Space Station, that we will do biological research there as well. And it is likely that at some point we're going to synthesize DNA on the moon. And we're going to do other biotechnological things that we do on research labs in the world here on Earth. So there's always a dual use considerations that what if this can be misused. So we need to have at least some frameworks, some protocols for this, and this is absolutely in existence so far. NASA just released about a month and a half or so ago their moon-based plan. There is no mention whatsoever of anything related to biosafety, and least of all biosecurity. There's a little bit about how to handle experiments going from Earth to the moon, but this is vague and it does not specify biological samples. But there are some planetary protection considerations at least. And then, of course, there's also the possibility of forward and back contamination going specifically with the planetary protection thing. So now, diving deeper on the moon. So the moon is a new operational domain. So lunar habitats sit beyond existing analogs. So if you don't know, there are many places on Earth, or not many, but some places on Earth where you can try and mimic the space environment when it comes to psychological factors and the ecosystem factors as well. So the main ones here that I have are the Antarctic stations. So, for example, Concordia, which is operated by France and Italy, harbors many European Space Agency researchers, and they just get isolated in Antarctica, in the middle of Antarctica, in the middle of nowhere with like minus 60, sometimes minus 80 degrees Celsius for like 6 or 7 months without any sort of resupply missions or anything else. So here an evacuation can be only done seasonal if they're deep in winter. If they're deep in it's going to be very dark there and everything. It's just like it's impossible to get them out of it in due time if something goes wrong. Resupply can take months. There's a partial atmospheric isolation. They can still open the hatches and get some fresh air, even though it's minus 60 or minus 70, but you can still do it. And there is onboard bioresearch that is existing, but limited. It's mostly focused on astrobiology and trying to understand how the Antarctic environment can mirror some potential ecosystems or biospheres outside of the world. So what researchers do there sometimes is that they go out, they collect some field samples of the ice cap, and then they sequence it. They analyze it in the labs within the stations. There's the submarines. ESA, for example, has some mostly psychological research going on with submarines to basically understand how isolation for long term can affect humans. So the evacuation is, in a way, some sort simple. You can just surface it or dock it to the continent. That can take a few days. Resupply can take weeks. There's a full atmospheric isolation if you don't surface. And there's no onboard bioresearch. So this is something that we can draw some elements from submarines to the moon. And then the ISS, the International Space Station, there's a lot of microgravity. Evacuation can take hours. This is something that is quite standardized as well. Resupply can take months. Atmospheric isolation is full, but again, there are some resets that happen once every few months with the resupply missions. And there's onboard bioresearch that happens per payload. And I'm going to talk about this very briefly, but there's a lot of biological experiments at the ISS, but they are analyzed on a biosafety level on a case-by-case scenario. Well, next slide, I'll talk about this a little bit more. And then for lunar habitats, evacuation can take days going back to Earth. Resupply can take months, but it's way more expensive. Atmospheric isolation is absolutely full, and onboard bioresearch is potentially continuous, autonomous, even with synthetic biology. Imagine just having a full biotechnology lab on the moon. That's how it's gonna operate. So it's surely going to be more developed than what we have at the ISS, even potentially something similar to what we have in Antarctica. So what current frameworks cover when it comes to the moon? I'm almost done, I promise you guys. So the first one is planetary protection, which we already covered. So there's the Outer Space Treaty, Article 9, and the Coast Guard policy that is quite standardized, and this absolutely applies to the moon. This is pretty well done. Then the other thing that is established is the human spaceflight biosafety focused on the International Space Station. So right now, we have the JPR, which is the Johnson Space Center Procedural Requirements 1800.5, which basically shows and delineates how biological research should be conducted when it comes to risk assessment and evaluation and biosafety perspectives to when somebody proposes a biological experiment to the ISS. So here's how it works. It's very simple. I submit to NASA what I want to do at the ISS with, let's say, Naegleria grouperti, which was what I sent in the what I sent to the stratosphere. Then they have a committee, which is the Biological Review Board, that is part of the Safety Review Board of NASA, and then they're gonna give a biosafety level classification. So they have a modified biosafety level framework that is paid by payload by payload. So it's a case-by-case scenario. So this is basically how it works. Every experiment is analyzed independently. And then, again, a main gap is that, of course, they don't accept any great pathogens. So the ones that are classified as BSL 3 and 4 on Earth do not fly to the ISS. So we cannot send Ebola to the ISS. But we can send, for example, let's say trypanosomas or salmonella, who are clear pathogens to humans, but biosafety level 2, because they do not disperse in an aerosol manner. But then we don't know how this is going to behave due to microgravity. I already explained this. And then the main gap here for Moon is having a habitat resident biosafety. Do we really think that having a payload by payload review process is enough for lunar habitats? I personally don't think so, because it's going to be a much more autonomous research environment than the ISS. And least of all, there's no biosecurity for in-habitat research whatsoever. Like NASA, ESA, nobody's really thinking about how to govern dual-use research on the moon. And then maybe this is a little bit, it's very frontier field, but maybe for like the next 10 years, this is not really relevant. But for the next 30, 50 years, this is now the time to start talking about this. So what transfers from Earth and the ISS? So there are main pillars here. So the agent classification, engineering, controls, personnel and procedures, and incident response when it comes to biosafety. So the terrestrial lab has already, they all have formal risk groups and biosafety level frameworks that we could definitely use on a lunar habitat. We have facility scale containment. So we have the cabinets to contain the flow of air. We have isolated spaces that should be also done on a lunar habitat, so facility scale, personnel and procedures. So we have formalized biosafety officers function, standard operating procedures, training, all of this. And we have full institutional escalation if there's an incident, and we know how to basically handle spillage, exposure, or escalation. Then in the International Space Station, we have what I already briefly mentioned about the modified biosafety level framework. We have a hardware level containment and a glove box. So each experiment is locally contained within a specific device, depending on the risk assessment conducted by the Biosafety Review Board of NASA or ESA as well, depending on ESA has was delegated to NASA to have some more authority on the matter as well. This talking about the ISS. I generally don't know how the Chinese do it on their space station. And then we do have, again, for personnel and procedures, a ground-based biosafety review board, a per payload review, and mission training. And then for incident response, we do have onboard containment protocols and emergency return protocols if needed within the ISS. So for the lunar habitat, what I believe that would work well is a hybrid framework between what we have in terrestrial and ISS labs, and further adaptation needed for the continuous habitation of the moon and the microgravity implications, understanding that the lunar regolith can have impact on the samples and so on and so forth. For engineering controls, we have the habitat integrated biosafety architecture. That is something that I strongly recommend space agencies do, that instead of just doing it on a reactive manner, they should imbue it on the architecture whenever they are just assembling and building and designing the habitat, like basically the modules where people are going to stay and conduct the research. And then we should have on board oversight role, delegation needed, and this is largely undefined right now. And then incident responses, we should have autonomy dependent protocols, and for now it is constrained, so we need clearly to have adapted protocols and standards for this. Then finally, biosecurity, where Earth tools haven't yet transferred. This is not related entirely to biosafety, but again, as missions move further from Earth, ground lab oversight gets harder. So they have to be more autonomous the further we go from Earth, every mission and every research that we can conduct there. So the main gaps here, I'm just going to ask questions. And the main gaps that we have here are, first, who has oversight of biological research conducted by international crews in commercial lunar habitat? So let's say that SpaceX has its own habitat there. Who has oversight of this? SpaceX, the United States of America, NASA? Who has it? We don't know. We just don't know. Nothing standardized about this right now. How does dual-use research review work 384,000 kilometers from any institutional biosafety committee? What screening applies to biological materials manifested for lunar transport? So whenever we bring something, a biological sample from Earth to the moon, how do we basically assess that this is dangerous and it should go or not? And then should we implement chain of custody and access control for in-habitat and return biological samples? Should we basically limit who has access to those samples, who cannot go there, who cannot touch it, who can? Should we do it? And then, of course, also putting the IBBIS flavor here, most importantly, is what governance applies to engineered organisms and in-habitat DNA synthesis. So if we have a synthesizer, a benchtop synthesizer on the moon, are we going to screen or not? What can be screened and what cannot be screened, and how are we going to make sure that somebody does not misuse this whenever they can? So why now? Because requirements built into design are cheap. As I said, biosafety and biosecurity requirements integrated during the design stage become structural, while doing it reactively make them fight the hardware, which is something that historically aerospace agencies have done recurrently. They say, "Oh, we want to go to the moon. We want to go. We want to launch a satellite," and then they just tackle the problems as they appear. And right now we can finally anticipate a big problem. And then terrestrial disciplines have decades of standards work. So we do have the ISO 35001 that focuses a lot on biosafety. We have the World Health Organization standards for biosafety levels management. We have the International Gene Synthesis Screening Framework. And of course, we have the Biological Weapons Convention at the United Nations, which is the main governing body for dual-use research and purposes of biology in general. And another argument is interoperability. So lunar operations will have more actors than the International Space Station. So without shared rules built early, each defines its own regime and operations fragments. So for example, NASA and all of the signatories of the Artemis Accords are focusing on the moon base. China and Russia are doing their own moon base as well, which is called the ILRS. And at some point, I'm pretty sure it's going to be useful for them to just have standardized and interoperable, let's say, equipment and telecommunications frequencies, and in this case, also biosafety and biosecurity standards as well. We may need, going back to the rescue agreement of UNUSA, this is something that may be useful in the future. So this needs an interdisciplinary working group, so space agencies, biosafety and biosecurity experts, and commercial lunar operators. So my job is trying to basically bridge the gap between engineers and biotechnological researchers and biosafety and biosecurity experts, trying to make this a little bit more constructive. With that, I thank you so much for listening to me. I know it's been a very extensive talk. I am happy to take on any questions you may have now. And that's it. Thank you so much, guys.
Thank you so much, Lucas. That was fantastic. I think that it's so interesting about your work about what you have been telling us, especially at the intersection of what you were of what you were saying about ABIS and the IGEM space. I mean, showing that this is not just a science fiction idea, and that is something that people thought that it's going to be the future, but right now it's currently being the present right now. So and I really resonate with one of your ideas about as we start sending living beings and living biological matter into space instead of just sterile metal or satellites or things like that. We really need to work on a new playbook or a new or new concepts for biosafety, biosecurity, and space law. So with that in mind, let's open to the audience. Drop your questions in the chat or raise your hand if you'd like to ask something to Lucas directly. In the meantime, I think that I will just ask one question myself since I'm very curious. So I
Mhm.
mean, Article 9 of the Outer Space Treaty requires, like as you have said, avoiding harmful contamination like forward rivers and such. So, however, with SynBio, as you know, often involves modifying genetic organisms, more or less like I wouldn't say on site, but it would be the ideal thing for synthetic biology, especially with in situ resource utilization. I mean, it's on the name. So how do you think that the legacy or the playbooks should be updated, especially in space treaties, to distinguish between intentional biomanufacturing, we would say, or intentional symbio, and uncontrolled forward contamination?
Great question. So I think the main answer to this is that it depends on where we're bringing the. This is my personal view. Okay, it's not necessarily like the right thing to do, but it's basically where we are sending the organism. So, for example, if we go to low Earth orbit, if we have the International Space Station, they already have the safety biosafety protocols that already work quite well. It's on a case by case scenario, but it works. I'm like, if it works, who am I to judge? But then if we go to deep space, if we go to the moon, for example, or Mars, this is absolutely different. So I think we just need new treaties. I don't think maybe I don't know how it works when it comes to amendments. When it comes to these treaties, I don't think they happen. But also, UNUSA has some other principles. So maybe we could start by having principles which are legally non-binding. So the treaties are legally non-binding, but the principles are just like, hey, let's have interoperable telecommunications. This is something that they do this, but it's not mandatory. But maybe we could start with this on a UN level. But I think most importantly, it is just discussing with space agencies and governments and making sure that we have, I think, dedicated frameworks for that. I think it requires dedicated frameworks. And then, of course, it goes to the biggest challenge of making sure that the nations just agree with each other. Right now, it's always been like this, but right now, in particular, there's a big geopolitical tension between the United States and Europe mostly, and Russia and China. And this is already entirely being reflected on the lunar landscape with both sides having their own moon base plans. So we'll see how that's going to go. But that's how I would suggest doing it. I think it would just have to sit down everybody together, and I think the UN is a good place for this because it's where most like everybody. It's one of the very few tables in the world of space where every stakeholder, every major spacefaring nation is present and has a voice. So maybe this is a way.
Thank you so much. I don't know if anyone has got a question, or I'm going to check in my private part. So let me see. Okay, so I've got a question on private sent by Luma. So thank you for the talk. I'm very new to space. So you say that you've been working closely with students through iGEM who are designing incredible space biotech solutions right now. I am a teacher at MSP, and given my students and young researchers are moving faster than UN committees or space treaty negotiations, how can we still instill, a culture of biosecurity responsibility directly into the developer workflow instead of focusing only on the lab?
Also, very good question. And for iGEM, this is already, I think, done pretty well with the responsibility program. So every iGEM team needs to have they have to fill their safety forms, right? So this is something that they already have to think about not doing. There are clear rules that the IGEM Safety and Security Committee stipulates that teams are allowed to do things or not. So like for IGEM, I think this is great. For the other things, it is, I think, again, it's putting this in the framework. So one reason, for example, that one thing about the Outer Space Treaty that we could mirror when it comes to potentially adopting these new frameworks dedicated to life sciences applied to deep space, for example, is that there's a good reason why uh, nations do not store nuclear weapons in orbit, and it's the Outer Space Treaty. It says, outer space and the Earth orbit must be used for peaceful purposes. Same for Antarctica. There's the Antarctic Treaty saying that this should be done on a peaceful level. So if we do this and then just delineating all of the risks associated with it, so that the accidental issues do not arise, I think that's the way to go. So this is something that the world has already done before, and we can totally mirror that.
Thank you so much. I've got another question through Luma. So it says, so if a private space company wants to fly engineered microbes to Mars next year for a mining project, is anyone actually stopping them, and how would that work?
I don't know. We don't have frameworks or anything. I don't think nobody's stopping them. If like SpaceX wants to, try the Starship on Mars and then send some I don't know, some cyanobacteria to biomine cement with Martian regolith. I don't think this is. I mean, it's not legislated at all. There's no framework. I think maybe NASA would maybe push against it in a way, but I think it would be more like informal, of some sorts. So this is one of the problems that I've been talking about, that we need to have more safeguards for this.
And that same person asked that if an Earth lab mutates on a synthetic strain under radiation. So I think that this person wants to say is if something on Earth went to space and mutated under the radiation and brings it back to Earth, who is in charge of checking if it's safe, and would that be considered back contamination?
It would not be considered back contamination because the organism originally came from Earth. So it's just a modified strain of something that has terrestrial origins. So back contamination is whenever we have like an alien bacterium on Mars and then we bring it back. But it's a good, let's say, gray area there. And right now, the main organ that would institution that would oversee this is the operator of the mission. So, for example, if it's a NASA operated mission, it would be NASA, at least originally, and then NASA may recruit some experts from universities or some researchers if a more thorough evaluation is required. But they already have some containment facilities for this. They already have some protocols, at least for the ISS when it comes to this. So it would be the space agencies mostly. Same for Roscosmos, I believe for the Chinese National Space Agency as well. So it would be them for sure. They would be the front line of defense.
Okay, thank you so much. I don't have any more questions through Luma, but I've got one question myself again. So if you could change or fix just one single rule on space law today to make room for SynBio, what would it be, or how would you make room for synthetic biology, or what would be one thing that you would like to see happening,
Yeah, what I would change, immediately, what I would try to change is that I would try to amend. It's not a law or like a treaty, but I would try to amend the moon base framework from NASA, because they don't mention anything about this, and I would start with that. So I would just try to have like a whole potential paragraph or section on biological experimentation requires its own attention. Immediately that's what I would do. And then what I would need to change, I think, on a greater level, is that engineers they don't hear about this, and like we need to open channels of discussions between these two worlds, between engineers, people who are generally focused on like propulsion systems, on hardware, and like orbital mechanics, and people who has microbiology experiences, who has biosecurity experiences. So what I wanted to change is have a specific forum. It can be at the UN, it can be someplace else, to have this better synergy. So NASA does conduct, ESA as well, they do conduct, and they do oversee a lot of biological experiments and research applied to space, but it rarely focuses on genetic engineering or synthetic biology. It rarely focuses on that. There's a lot of like space omics, space health, which is great. It's amazing. Absolutely keep it up, but again, SynBio is another field that is something that we will do if we are going to inhabit Mars in the longer term. It's going to happen. It's there's no other way, and we need it to actually put people there. So uh, so yeah, it's something that it's extremely overlooked, and I would change on a global scale, having these two worlds talk more.
Yeah, I completely resonate with that, especially because when you're looking for bibliography or for new information, you can see that whenever you put down space science, you can see maybe 85% of the resources being pure hardware or pure software instead of also biological compounds, which at the end will be like one of the main tools that we have to make humans a multiplanetary species rather than being bound to Earth. So there are no more questions in the chat or from Private Luma. Thank you so much, Lucas, for being with us here today. It has been super exciting having you here, and as well in a personal part, to see you again. It's always exciting and always a pleasure to see you. And thank you so much for giving us your time. Sorry for being a little bit over time, and thank you so much. It has been incredibly interesting, and we look forward to having you in the future for more opportunities.
Absolutely, just feel free to reach out. I have my LinkedIn, I have my email. Just reach out. I'm happy to help. And yeah, also good luck with a space village. Also, if you need anything, let me know. And yeah, it's fun. It's exciting times. Yeah, it's lots of work to do, but exciting times. Yeah. Thank you, Yasin. Thank you, everyone.
Thank you so much. See you very soon.